Okay, so shalom everyone, and welcome to our first webinar of 2025. My name is Yoash Dvir. I'm the CEO of Technion Australia. Technion Australia is the local society of Technion, Israel Institute of Technology, and we are here to raise support to Technion's activities and research, and we also run local programs like this webinar, meetups, school programs, and more. Tonight, we are privileged to host Professor Shai Shen-Or, who is a professor at the Faculty of Medicine at the Technion, where he heads the System Immunology and Precision Medicine Laboratory since 2012. His research laid the foundation of CytoReason, a company who is building a computational disease modeling technology, which it applies to further drug development in collaboration with leading pharma companies. In addition, he is the co-CSO of the global nonprofit, the Human Immunome Project, and oversees its scientific design protocols. This is a whole new use of AI for healthcare. These efforts, applying computational science and information technology to biology and medicine, could advance the diagnosis and treatment of immune-related diseases. With a background in informatics and computational biology, Professor Shen-Or is the director of the Zeeman Institute for AI Solutions in Healthcare at the Technion, whose goal is to ensure academic innovation in the field makes impact at the real world. He's also the co-director of TechAge, the Technion's AI umbrella organization, and he's the director of the Technion Institute for Healthy Aging, founded recently as a multidisciplinary institute set on addressing one of humanity's largest health challenges. Professor Shen-Or received his BSc from the Technion in Information Systems in 1999, an MSc in Bioinformatics from the Weizmann Institute of Science at 2002, a PhD from Harvard University in biochemistry in 2007, and performed his postdoctoral studies at Stanford University. His research has been cited numerous times and has been featured in system biology textbooks for students. We will allow, as I said, Q&A at the end. Professor Shen-Or, the stage is yours.
Thank you, Raj, and thank you for inviting me to this webinar. It's a pleasure to speak to folks at Australia and Technion supporters. So what I'm going to do today is tell you a bit, I think, through the lens of my own, starting from the lens of my research to kind of, I guess, how I've evolved as a researcher and the things that have interested me, and also the process of that is going on at the Technion in terms of how do we think about human health and kind of how universities plays a role in this, and some of the directions that we're pushing in that I happen to be involved in. So I guess, as Yoash said, I come from a background of informatics, but I fell in love with biology and healthcare. And so starting, I guess, for my master's, I did my undergrad at Technion. I started from a master's studying E. coli. And to give you a sense of this, the E. coli genome is 4.6 megabases. I was studying it around 2000, and this was a sequenced genome, and we could understand what's going on to some extent. It's still a fascinating organism to be studying. And post completing my master's, I decided I needed a challenge of a multicellular organism, and I moved to the worm C. elegans, which was actually the first sequenced animal. So just to give you a sense, the genome of E. coli is 4.6 megabases. I started studying C. elegans in 2002, and its genome is 100 megabases, right? Significantly less than you store on your phone today. And we understand C. elegans a whole, depending on a half glass full, half glass empty. You can say we understand it, great as an animal, but actually less so than we understand, the bacteria E. coli. And then I shifted to studying humans, and the genome of humans, again, to give you the sense of scale, is 3 gigabases. Particularly, I've been focused on the immune system in humans. And since we care, about our own health, we've been generating a lot of data in humans, and yet the complexity of humans is such that we actually understand less than we do about C. elegans or the bacteria E. coli. And what's been changing recently is that we now have the technologies to actually sequence and measure many different types of biological layers in humans, and the data has been exploding. And there's some glimpse that we can actually change the equation here on the understanding, so that suddenly it's not just less, we can actually start understanding more. And as you see this, my own personal journey, I think you can start imagining how computers would fit in this, because there's more and more and more data to actually make sense of, and you would struggle to do this without actually putting a lot of computer science and machine learning and artificial intelligence in. So to give you, kind of how I view this from a global scale, and I guess through the journey that I've been going through since the mid-90s, it's actually biology, as many other fields in science and in other walks of life, the data has been exploding in the past 20, 30 years. But the problem is that getting insight, getting value from the data doesn't follow the same trend. So it's growing too. So that's the half glass full, I guess, but at a very different rate than the data exploding. Data can be exploding exponentially. Insight grows linearly. Basically, every day the percent of data utilized drops. And this is an issue if we actually, this is expensive experiments, we want to get out of them. We want to get as much insight as we can from the data, and yet we're struggling to do so. And so that's exactly where computer science fits in trying to develop methodologies to get more. And I'll just give you an example. Think about the difference between working on animal models such as mice, where people often study them for, because of there's no way to do some of the experiments in humans, and we're trying to get cures and so forth. But obviously, mice and humans are not the same. The last common ancestor we shared is 400 million years ago. They live in inbred. They're inbred. They live in clean labs. We don't. So there's a lot of gaps between us. And as an example, we developed an algorithm back in 2018 that basically said, took all the data that's out there and taught a computer to help us translate what of the results that we see in mice would make sense, would fit what we would expect to see in humans. So this is an algorithm we call found in translation, was developed by a PhD student of mine. So if you think about, normally people take mouse results and they want to understand what's going on in human, say, oh, I got the results in mice. Let's see, I'm going to assume that things in human are the same. Obviously, this is a big issue. And in fact, most, we've cured every possible disease in mice. But that doesn't necessarily help the humans. Most drug development that works in mice fails in humans. But our machine learning model learns about using all the publicly available data about human disease and mouse models of human disease. It learns how to translate better, so that we give it a new mouse experiment. It knows how to fill the gap better, and we can do up to 50% better than the standard translation. So it's an example of how computer science fits in trying to make sense of data in biology. And in fact, if you want to extrapolate this further, think about the process that we need to be going through in those past 25 years. So just to give the equivalent, we moved from phones, being analog and not really having any kind of information storage, to like these Nokias that collected data but it wasn't useful, to having smartphones that are collecting data that becomes extremely useful, such as Waze, such as maps and others, where they're so useful today that if we forget our phone at home, we run back home and pick it up, right? Sweating, our sweat glands are stressed out when we forget our phone. And what we're trying to do in biology, during the same 25 years, we moved from biology that basically had no big data, microscopes, people looking at images and writing in notebooks and so forth, to being able to generate big data, as I kind of was giving you the example with the different genome sizes, to the fact that actually to make sense of that data, to get the utility out, we need to basically build a model of what biology looks like to get the same equivalent of what we see in the smartphone. But this is way more difficult, and we'll touch on it a bit later in the talk. So where am I focused? I'm focused on the immune system. So you can think about the immune system at a very high level as a sensory system. This is a system, it's almost like a sixth sense. It's responsible to monitor your external environment and your internal environment, and to respond to this so that ultimately your body maintains what we call homeostasis, so a balance. And as it does so, this is a learning system. As it does so, it keeps changing to adapt to the environment. So really like a sentinel, if you like. And we study the immune system, and particularly when we studied it in humans. As we all differ between each other and we have a different life history, and the criticality of the system for medicine, we often think about the translational or applied research that we do in the lab as being as trying to answer this very simple question. Imagine a person walked into a physician's office and said, "Hey doc, how's my immune system doing?" Right? Sounds like a simple question. Actually, we don't really have a good answer for this. We don't understand how the immune system varies within an individual over time, and we don't understand how it varies between individuals. And if you want to compare this to cardiovascular health, if the patient asks, "How's my heart doing?" the physician has a lot of different metrics to assess cardiovascular health and intervene either with lifestyle or with therapies or surgical interventions. For the immune system, that's not the case. And in fact, if I look at COVID, it's a good example, unfortunately, of the lack of understanding that we have of immune variation around the globe and what it means. So what is the physician thinking about? Well, first of all, and why is it saying no clue? Well, the problem is that this is a distributed system in which the units are cells. They are the quanta unit, the atomic unit of the immune system. And in fact, there are many, many different cell types, if you like, that each of them has its own flavor. And you're trying to understand really what the kind of the big picture is, so that because all of these different cell types, each with its own function, together they yield this emergent phenomena of immune response. And what has changed in the past 25 years is that it used to be, I'd say I'm going to study the immune system in humans, and this was basically thought to be crazy sci-fi idea. You can't do this. Humans are too variable, and so forth. To having the technologies that give us actually the big picture. So in this case, this is Mona Lisa on coffee cups. And in fact, if I remember correctly, this is a picture taken in some festival in Australia. So yeah, people might know it. I gave a talk at TED, and I had to get a license from the photographer who took this to display it. So yeah, appreciation of good coffee, I guess, and the emergent phenomena that comes from thinking about taking all the different components, all these different cells, and suddenly seeing the big picture of what immune response is like. And so what we now have are these types of technologies, and my lab has been both developing these technologies and using them to study humans to understand how our immune system varies over time. And particularly, we've been looking at a process that we think of as immune aging. So we've been tracking a group of 150 humans over the past 17 years. This has allowed us to make sense of what your immune variation looks like. You have all these different cells. We can now measure all of them together. They don't vary randomly. There's actually a very predictable pattern by how your immune system cells are going to change, especially in healthy individuals. And we can now track this and describe basically a single road or a single trajectory, if you like, by which all people, as they go through life, travel and their immune system changes at the very high resolution of many, many different immune cells. We can measure this in your blood, and we can tell you where are you on the road. And we call this image for immune aging. We can measure it by cells or genes and so forth. So basically, it's the position you currently have on the road. And while we all travel the same road, we do so at very different rates. So some of us may be traveling in a Ferrari, and some of us be traveling in the bus. And I'm happy to tell you afterwards what we think about which is better in which conditions. But basically, it's a conserved pattern that, as far as we can see right now, and we keep studying it. We see it conserved around the globe. It's in healthy individuals. This is the largest variation of the immune system. We're not, though I'm using the term age, and you can think about this as being something that's predominant in older adults, children in Bangladesh who are 3-year-old have an immune system that looks like a 40-year-old person in Palo Alto, California, as an example. And both children in Bangladesh and old people in California don't respond to vaccines because we are looking at an immunotherapy, yeah, because our immune system varies. And what happens when, say, we give a vaccine is basically to ask, if I'm here on the road and we now go off on a ramp, will we all reach Rome, right? Will we all reach our destination, or each of us is, some of us will respond to the vaccine and reach our destination, and some of us will not, right? So especially in the cases of immunotherapies, some of the latest, most successful drugs in the market, where we're seeing a huge variation between individuals and how they respond to the treatment or not. As an example, the recent a couple of years ago, the immune checkpoint inhibitors were awarded the Nobel Prize. These are revolutionary drugs that have changed how we treat cancer, but we're talking about somewhere between 20 to 30% of patients responding to them in a disease such as melanoma. And what we're showing is that by tracking how your immune system changes, this actually allows us to identify predictors of whether you'll respond or not to those treatments, because we understand the variation that exists between us. So that's kind of a sense of kind of how we're looking at our own research and the variation that we're using these technologies to measure the immune variation. But it also, I think, reflects this idea that humans vary a lot, and especially with age, and that ultimately led me to a place where I was looking to see, well, how does the variation of our immune system, and particularly as we change over time, how does that relate to all of the other physiology, all of other kind of systems, given the centrality of the immune system? And long story made short, about 4 years ago, with 5 other faculty at the Technion and different faculty members of Technion in different different departments, we submitted an application to the Technion leadership to set up a Healthy Aging Institute. Took a couple of years, but basically this past June, the university authorized to set up a new research institute at Technion that's focused around healthy aging in a very multidisciplinary manner. And I'll just give you a bit of a sense of, and then obviously this is connected to the research we're doing on the immune side, but give you a bigger picture of why we think this is a grand challenge that we are particularly suitable to addressing. So there's, you can think about human aging today as a grand challenge, and we have unfortunately, we have I guess the buy-in or the support of institutes such as the U.S. National Academy of Medicine, who 2 years ago said this is the largest problem that exists today in healthcare, the difference between lifespan and health span. So healthy aging, the difference between when we die and when life actually starts deteriorating and it's no longer as fun as it was, right? That difference today sits on average about 8 to 10 years. It varies based on social economic status. So even the richest people in the world suffer from this situation. But at the poorer side of social economic status, you're talking it could easily go to 15, 20 years. And that gap is where most of economic healthcare costs go, right? So the largest percent of effort of healthcare costs are going in the last decade of life when things are no longer fun. And so you can think about clinical medicine as something that's been extending life, but not necessarily its quality to the same extent. And so this is what the National Academy of Medicine said, this is the biggest challenge we need to solve. And the reason we need to solve it is not only because medicine is basically unsustainable without solving this gap in quality of life to end of life, but also because there's a more global phenomenon that's happening, and that's the world is actually becoming significantly more mature or older than we're used to. So just to give you a sense, today, if you go around the globe, the statistic is that 1 in 9 people is above the age of 65. In 5 years from now will be the first time there are in the world that there are more people over the age of 65 than children below the age of 10. In 25 years, we're talking in numbers where 1 in 4 people is over the age of 65. So this is a huge shift that has an implication that goes way beyond our own individual lifespan, health span issue, but also means the way we have our society set up now is actually not a way that's actually sustainable for the future. And we need to figure out how do we bring the dividend, if you like, or the contribution of older individuals into society in a much more significant manner, not only because they're a much bigger component, but also because economically this is unsustainable. And so solving this gap is something that both the National Academy of Medicine and the World Health Organization have recognized as being, as big problems. I personally think about it, it's as almost as big as climate change, or maybe second to climate change in that respect. And I will also, to give you another kind of sense of the change, this is not just a shift in kind of in the world in terms of the demographics of young to old, but also where's things happening. So if today the most populous, the fastest growing country in the world is Nigeria with the highest birth rate. In 25 years, the number of children born in Nigeria every year is equal to the number of children born in all of Europe every year. Okay, so the world is changing, and we need to start preparing to actually be able to address this. And there are already countries in the world where the society is shrinking, such as Japan. Every year, less 100,000 Japanese in the world because the birth rate is significantly smaller than death. So why are we thinking that the Technion plays a significant role here? Well, because we believe we're uniquely situated to impact human health. As you may know, the Technion's president, Uri Sivan, made a strategic decision when he came into office that the Technion will invest in particular directions. There's three such directions. One of them is human health. The second is sustainability, and the third is digitization of industry. So human health is a huge commitment that Technion is making. We've made revolutions in other things such as defense, high-tech industry, agriculture, and we believe that this is the place we need to be investing next. This is also, by the way, a decision that's been made by the Israeli government that's thinking about the growth of Israel for the future and saying that the investment needs to be in healthcare to actually be able to grow as a country. We believe we have a unique differentiation here. Not only do we have a world-leading technical university, but it also has a medical school and affiliated hospitals that actually give us an edge in being able to address this. We've reviewed all the healthy aging institutes that we've found around the world. We believe we have a unique offering that I'll tell you about that we think can give it a significant contribution to this important grand challenge. We already have a huge amount of kind of core capabilities and excellence of researchers engaged in doing studies around human health and healthy aging. And really, this is an institute that would bring them all together. And we're sitting in Israel that has a top-class healthcare system that's tightly knit and can execute quickly, which we believe can serve as a beta site for many different types of interventions that we can then export and inform the world of, just as we did, if people know, during COVID, the vaccines that were given in Israel, and Israel was the first country to vaccinate an entire population. We used our very significant electronic medical system, electronic medical record system, to inform the world on what vaccination does and how to think about populations at risk. So this is kind of why we went into healthy aging. The Healthy Aging Institute consists of 4 components, and those includes what we call iTechAge, which is the mission control, and we're recruiting faculty in this field. Systems Medicine Center, which is an umbrella to all our core facilities to basically allow us to lower the energy barrier needed to go into this field and make revolutions. It's allowing us to test technologies and so forth. There's a direction of interdisciplinary research themes that we're studying across the board, and I'll review in a second. But really what is unique about this is that we are developing or we're putting together a human monitoring center that's a high-end kind of facility to measure to phenotype or to kind of measure humans in many different ways in a comprehensive way. And we are going to be putting a lifespan cohort, individuals that we're tracking over time that in this human monitoring center that are a shared resource for the entire university to be studying healthy aging from many different angles. So what does this look like? We're talking about, say, 1,000 individuals is how we're thinking right now that are tracked longitudinally. They're being tracked in using multi-angled measurements, so the molecular, cognitive, functional assays over time with monitoring the environment and tracking clinical phenotypes. This is, as I said, going to be a centralized resource, and we're building it in a way that it's the top of a pyramid of much larger cohorts that we have less information on. So we've been partnering with HMOs, with medical organizations and hospitals and so forth, including in Israel and in the world, where we can use our very comprehensively profiled cohort to inform larger populations, or the reverse, study something in a large population level and try to understand its mechanism on the top. So this is the lifespan cohort. The idea here is that the mission control iTechAge really kind of specifies the design and the execution of the lifespan cohort. We're tracking it in the Systems Medicine Center, and that data goes to the research teams to actually study. We are issuing seed grants to faculty members and clinicians to basically drive challenges. For instance, we just put a call for proposal. We got multidisciplinary proposals across the university for different issues around aging, as well as driving ideas like, how can we define in a quantitative measure stress, right? And other measures. The idea is that the research teams, as they produce results, we're using this mission control to actually impact policy by partnering with governments and healthcare organizations in Israel and beyond. So this is the design of the center. We have now about 60 faculty members who've already signed up across the university to play a role in this, giving you the breadth of what this comes. This is not just, though healthy aging, you can think about biology and medicine, but it also includes people from electrical engineering and a lot of faculty in architecture, thinking about urban planning and design of buildings that are fitting for older individuals, people from biotechnology and food engineering, or designing foods and so forth, chemistry for drugs. So this has a very broad coming together of synergism across the university. What are the research teams about? Here's an example. We basically designed it as having 7 research teams. They go from cell mechanisms of aging to thinking about systems aging, people like myself studying the immune system, or others studying the endocrine system or the cardiovascular system. We're looking into, we have a whole research team dedicated to brain and cognition, a research team around artificial intelligence and diagnostics of biomarkers of aging, personalizing foods and drugs, assistive technologies, robotics, and so forth, and community, thinking about the population and how they come together. So the breadth of the institute we're putting together really kind of goes from in 2 axes. On one hand, it goes from the micro, from the cell, all the way to the population, and on the other hand, it goes from basic discoveries that are sitting on the left-hand side here that take often many years to mature into new drugs and interventions and so forth, to directions that are more engineering based and clinical based, where we are hoping that the basic research informs them, but they can have an impact tomorrow on people's lives. And I'll give you a few examples of such research teams just to give you a sense of how the breadth of this institute we're putting together. So I'll say the idea is we monitor the humans, we identify mechanisms, we are using a variety of different models to establish causation, understand the mechanism, and then kind of suggest interventions and so forth. So we have a whole path from discovery to intervention. We have many faculty members involved. So here are each of the research team's leaders, different research team leaders that are actually playing a role with this, some in biology, some in architecture, and different faculties. Every research team has 1 or 2 faculty members that lead it. And here's a few examples. So this is, for instance, the research team around brain and cognition. It's led by 2 faculty members, Firas Mawasi from biomedical engineering. He works on humans studying cognition and motor functions, and Ben Engelhard, who's a assistant professor in medicine, and he works on learning in animal models. And the research team, which involves over 12 faculty members, is studying why does brain function decline with age? how can we reverse this process? So the idea is to try to decipher neural mechanisms, discover biomarkers of aging, and ultimately come up with interventions that are either coming from the animal models or they're coming from the humans. So this is an example. Another example on the engineering side is Uri Lesmes. He heads our personalized food and drug delivery research team. Uri is also a member of the Technion Sustainable Protein Institute that we recently, Sustainable Protein Center, recently put together around food engineering. He works a lot with industry, and what Uri does is design food for older adults that has the nutritional supplements needed in them that are actually lacking with aging, so that he can improve muscle strength and other factors using better foods that are healthy and are basically replacement formulations to things that are on the market for the average population. That's another type of engineering feat, and Uri can be fed by research that's coming from the basic science and what needs to be in those foods. Here's another example. Ariel Fisher is an assistant professor in biomedical engineering. Many people involved in her research team, but I'll just give you an example. If you're looking here, this is a couple that's walking on the beach, and the woman is wearing a knee brace that Ariel designed. This is a dynamically stimulating knee brace. It's a knee brace that actually dynamically stimulates the muscles as the person is walking to generate the best response for kind of the best, most stable and active walking. It just passed clinical trials at Stanford University, and we're thinking about this as here's an example of things that we can personalize and further beta test on our cohorts. And last example I'll show is on a population level, and this is a research team that's the community research team that's led by Merav Aaron Gutman, who's a faculty, he's a urban sociologist at the Faculty of Architecture and Town Planning. And what you see here on the right is a picture of the Hadar, it's a bird's eye view of the Hadar neighborhood in Haifa, where every building that's painted in red is basically estimated. The intensity of the red is an estimation of the percent of lonely people living in this building. So Mirav built a whole data science model that incorporates many information that's coming from Social Security in the city and so forth, obviously all de-identified, predicts loneliness. She verifies the model based on the number of days it takes to actually identify that a person has passed away. And then she works with the Haifa municipality, with the social welfare department to better do city planning for buses, where to position community centers and so forth, so that the lonely people actually reestablish their connections with friends and so forth to actually change the dynamics of age. And what we can allow now in one single institute is to both study such kind of global population level, community level directions such as Merav studies, but also from the same people, ask them to give us a blood draw and study their immune system age, or other features that are interesting for faculty around the Technion. So I hope this kind of gave you a breadth of what we're trying to put together in this new institute at Technion and the importance of this. I'll say that all of this also has another arm, which as Yoash mentioned, I also co-direct TechAI. TechAI is the the umbrella institute for artificial intelligence at Technion. We're one of the leading research universities in the world around artificial intelligence. And as you can imagine, all this data that we're collecting has a huge amount of implications that require artificial intelligence in all its shapes and forms to actually make an impact. And so just to give you a sense here, TechAI's actually Technion is ranked #1 in Europe for the past 15 years around artificial intelligence. We include over 160 faculty members, 50 of which are basically developing core technologies in AI, covering all the aspects of modern AI, and really working in an interdisciplinary way across the Technion faculties. And in particular, we work across those 3 directions that the president defined as the strategic directions that the Technion needs to be focused in: sustainability, industry digitization, and human health, that I just kind of give you a sense of the Healthy Aging Institute. All of this is driven by the core AI that's developed at the Technion. So I'll say, we take the AI that from ChatGPT and so forth, and try to put it much more into helping directly society with safe and trustworthy assistance, companions, getting kind of things that humans cannot do well, and impacting science and engineering applications such as healthy aging. Here's how we're thinking about what we're doing in the healthy aging center, where we have this pyramid of people that we're tracking, as I kind of told you before. Those people are being tracked, will be tracked, or we're putting it together now by wearables, molecular level, clinical physiology, and so forth in a repeated manner. The data that's get collected, we believe we can now put into an AI model that's incorporating electronic medical records from Israel's HMOs, medical literature, and so forth, and ultimately build a version, a beta test of decision support that's going to guide those people. And again, the idea is that they're just the tip of an iceberg that we can then inform and develop the methodologies for. So in the, I guess, last 5 minutes, I'll go back to myself and give you a sense of how I've evolved in this. And I think another arm of the Technion is really how do we bring those impacts ultimately to society. And so for me, that really meant thinking not only about the immune system and how we age and kind of basic questions around these and diagnostics of that, but also how do we bring all this artificial intelligence that we've been developing in the lab, machine learning models to improve. I kind of showed you the mouse human, but there's many more. And how do we bring this ultimately impacting human health? And we were thinking around drug development, with the big question being, for me, how are we going to develop drugs in 20 years? Because if I understood that, I could build a model that would do this. And I don't think anybody has a good answer of how we're going to develop it, but my sense is there's going to be way more computer science and less mice. And so we started this process of thinking about drug development and building those models that I was showing you before. I founded a company called Cytoreason out of the Technion. The Technion kind of launched it. Today, Cytoreason is a 130-person company. It's actually the world leader in this space. And what it does is it builds an AI platform that's changing the rate by which we get insight from data in around drug development. And so it's basically building, same thing I was showing you about the Waze and the phone, it's building a computational disease model of disease biology. And with the idea that we need to be shifting biology from, from not very computationally using computers to something that's much more intertwined. I personally think biology changes dramatically every 100 years or so. Here's the 19th century, here's the 20th, but here's something that we need to be getting to the 21st century, and that's what we're trying to do, both in the lab and in the company. So with that, happy to take questions. Thank you for listening.
Thank you, Shai. I have a few questions here. I just want to mention that Professor Merav Aaron that you mentioned will come to Australia in May, at the end of May, and we will have an event with her here in Sydney, maybe also in Melbourne, but I cannot promise that. We will try. But definitely here in Sydney, we will have an in-person event with her. And just one thing before we go to the questions, Uri Lesmes that you've mentioned, Professor Lesmes, we had a webinar with him last year about his food technology, and you can find that in our YouTube channel if you want to go back to see what he was talking about last year. I have a few questions here. I'm not sure that I can understand everyone. Can you see the question, Shai?
Yes, I can.
So please, if you can.
Sure. I'll start here. There's a question from Maxine on in which city was the research done on the correlation between loneliness and urban planning? This was done in Haifa, in the Hadar neighborhood, by Merav Aaron, who's coming, I guess, in May, which is great to hear. And so she's been extending this now. She's been doing this now in Tel Aviv and so forth. And part of the Healthy Aging Institute is to basically do this across the population that gets studied in the Haifa area. So that's Maxine. And then I think there's two questions from Manuel. How do you compare glycan age and true age tests that are available in Australia? And will test be available in Australia? We're not yet in Australia, nor are we commercially available. I'm not familiar with those commercial tests that you're describing, Manuel. We've done comparisons with methylation age, which is like epigenetic clocks, which is probably the most common clock out there. And by and large, we outperform them or are complementary. So the two together give more value than each. There's many different epigenetic clocks, so some we outperform and some we complement. And I think in general, the field of biological clocks, if you like. So rather than asking somebody, how old are you? To actually look at how old are their different organs and tissues is something and systems is something that that's kind of a very growing field, and I think ultimately we'll end with a multi-clock. So there's going to be a component about the immune system, a component about different organs. I think it's a very hot field right now. A second question, I guess, was, are we planning to follow the samples in your study for the entire life? And the short answer is that the cohort that right now is going to be the Healthy Aging Institute. We're currently fundraising for it. And the institute is budgeted for 10 years. We will need to see what happens after. What we have shown in the cohort that we've been studying for immune aging for the past 17 years is when you measure people at high resolution, so rather than measuring 5 different cells of their immune system as you get in your complete blood count every time you go to the physician, and that's been measured in humans since 1957, when I measure 100 different immune cell types on a person, I can actually start stitching together people. So I measure a person, 4, 5, 6 years, and I can take another person that's 4 or 5, and I can start connecting the dots. So, while awesome to track them for their entire lifespan, how we do this from a financial perspective is not yet clear, but being able to build a trajectory of health across lifespan, if we design the cohort right, is something we believe we can do. And then I guess there's a question from Brian. Please discuss advances for organ transplantation. Less my field, so I can't, immunology advances. I can tell you one thing that we are doing. We are, with respect to immune aging, we are looking to see what is the effect of immune age on organ transplantation. So while we have shown that an old immune age can predict survival, has, people with an older immune age have poor cardiovascular health and where the immune side precedes the cardiovascular direction, we believe that in organ transplant, it may actually be the reverse, that a young immune age may be detrimental. Particularly, there are diseases in which the immune system attacks the body, such as graft versus host. And we have a study that's running now. We just finished collecting the samples, but we're still analyzing to try and understand the effect of immune system variability, and particularly around immune aging, with successive bone marrow transplant. So I guess I'll come again and I'll tell you. 2 more questions here, I guess. From Ricky, how would medicine research be passed on to various drug companies, and how would you decide which companies to pass on to? Okay, that's a big question. So I would say there are multiple directions. So obviously, the Technion is a university that's a nonprofit. There's researchers publish, and those results are open to the public. We do have a commercial arm. That commercial arm, either through licenses of patents or know-how and so forth, works with different drug companies. So potentially, if there was an invention made that wasn't made public, that was some publicly available publication, but also an invention made that was patented, that could be licensed to a drug company, and that would depend on the interest of the drug companies and where the Technion saw most, I think, most potential impact. There's a huge amount of at the Technion there's a huge amount of say for the faculty member in how things get commercialized or impacted. So it's my decision as a faculty member in the Technion to whether I put it in the public domain or do we patent and try to commercialize. It's my decision, really, ultimately, on who do we commercialize with, and so forth. So, Ricky, I hope I answered this to some extent. That's a loaded question. How am I measuring? Ruth is asking, how am I measuring immune age? We started measuring by measuring immune cells at high resolution. We have a machine that Technion purchased when I got here that cost about $1 million to measure many different cells. Now that we've identified a way of measuring immune age, we've developed way cheaper ways of doing it. We can now measure it on a standard, what's called a flow cytometer that sits in every hospital. And we're in the process of now scaling it so that we can do 100 samples a day. Right now it costs us $250 a sample, US dollars a sample to do, and we can do 30 samples a day, or 20 samples a day, sorry. We're now at a place where we very soon will be able to measure 100 samples for tens of dollars, and we're scaling it up to move there, and we can also measure it by genes or other ways. Oh, questions are coming up. I'm okay. Sue is asking, has there
We have two
been
more
developments
minutes.
until? All right. Has there been developments in telomere research with respect to reverse aging? There is a X Prize, large competition for $100 million for people who can rejuvenate you by 20 years. There's a bunch of measurements that are being directions that we're talking about. The immune side, we're actually considered right now the leading candidate to measure immune aging rejuvenation by X Prize, muscle and cognition. Telomere research, personal opinion, has been a bit stuck, but some of it due to resolution. So anyway, my opinion, I guess.
Shai, if you can answer
Is
this one,
there?
you promised to explain
Yes.
the
Which one?
aging journey. Is it better in a bus or a Ferrari?
Ah, all right. I promised, yes.
You
So it
did.
varies by the condition, right? Right. So it varies by the condition. So I kind of said it before, we already have quite strong associations, but having a high immune age is detrimental. So first of all, it predicts survival. It's a significant factor in predicting survival 7 years ahead. And we now know that it predicts particular types of cardiovascular functions, situations, and it precedes. So before physicians can detect cardiovascular deterioration, we can pick it up on the immune system already. And there are particular cardiovascular surgeries that we know we can differentiate outcomes based on the immune side. So there's certainly chronic diseases. It looks like an older immune age is bad, but as I said, we think that there are conditions such as bone marrow transplant, where a young immune age would be more appropriate, and so we're looking at that now. So generally, Ferraris are bad for your health, for the common diseases.
So just
All
one last question.
right.
No, I'll ask it.
Yes.
So what is the secret for healthy aging?
Sorry. What
What
was
is
the
the
question?
secret for healthy aging? That's what we're here for. Just give us the bottom line. How can we all do it?
Ah,
So what do you recommend, doctor?
I think there isn't one secret, but I think actually the way I think about healthy aging is this is truly a systems problem. And which is why we're tackling it this way. There's the loneliness, the social aspects, the genetic aspects, the biological aspects that are kind of relating to your life expectation, you know, general wear and tear, which is kind of why we're going with assistive technologies. I think no one's secret, but I guess live life well is as best as we can, and early diagnostic is an important component here. Yeah.
Okay, thank
Sorry
you.
to not have
No, no.
yeah.
So thank you very much, Professor, and everyone for participating today. If you have any other questions, please email us and we will forward it to the professor, and I'm sure he'll be happy to answer as much as he can. If you're interested in supporting the Healthy Aging Institute or Professor Shenor's research, please use the link that I just sent on the chat or go to our website. Obviously, all donations are tax deductible. And we have a health promotion fund, so the donations that we collect through there go to support this very important cause. Again, thank you. Thank you, everyone. Happy Purim, and see you in our next webinar. Thank you.
Thank you.